In this lecture, I will discuss sequence similarity search algorithms. Specifically, we will learn about BLAST, which is the popular sequence search method in bioinformatics, and we will also learn about some of the related tools. We have already learned that dynamic programming can be used to solve sequence alignment efficiently. This efficiency was quantified in computational terms as a time complexity measure. The amount of time that the algorithm would have to spend to solve this problem was proportional to the product of the sizes of the two sequences that we were aligning, because the number of times that we would have to repeat the process in a dynamic programming is proportional to the size of this table. In practical terms, if we are comparing two genes or two proteins, which would be several hundred to several thousand characters long, the sequence alignment would take less than a second to compute. But what if one of the sequences were really long? Say if we were comparing a protein sequence with the entire genome of an organism, which can be in the order of several billion characters. Or say we had a new protein sequence that we have discovered, and we wanted to know if any of the other species that we know of had a similar sequence. That would require we take our protein and compare it with the genomes of all the organisms we have available, which collectively would be in the order of trillions of characters. For the sequence search problem, the dynamic programming solution would not be as practical anymore. We may have to wait hours or days to find the best set of alignments or to find the best set of similar sequences from our database. I will call this problem the sequence similarity search problem. We have a short sequence that we are searching in a large database. If your database consists of a large collection of sequences, such as all of the proteins in an organism or the entire genomes of many organisms, you can just combine those sequences and think about the database as one giant sequence. We have already discussed a solution to this problem using dynamic programming, where the idea was to repeatedly find local alignments. But this again requires building the entire table, which would be too large and too time-consuming to do, and we would like to avoid that. Let's discuss some strategies that can find these local alignments faster. One of the popular methods to solve this problem is BLAST, which stands for Basic Local Alignment Search Tool. BLAST is a heuristic method. We use the word heuristic to indicate that a method employs a strategy that is likely to find good solutions or approximate solutions, but is not necessarily guaranteed to find the best solution. For most practical applications of sequence search problem, the accuracy achieved by BLAST is good enough. BLAST is based on the observation that if two sequences are similar, they must share short subsequences. Here we see the alignment of two sequences. The high-scoring alignment path aligns these identical triplets of characters. These identical triplets of characters can also be seen as diagonal matches in a dot plot. The idea behind BLAST is to, rather than building the entire table, we can explore only around these promising seeds. The dynamic programming extension process employed by BLAST is demonstrated here. Let's pretend that we have already identified where in the matrix we have triplets of consecutive characters. There may be other single character hits, but we are not considering them as candidate seeds. Starting from these seed positions, we calculate the dynamic programming table like we normally do, going both in forward and reverse directions. Except that when the score falls below a threshold, we stop extending beyond that point. Once we have extended all the way that we can, we then trace back to reconstruct the alignment path. This extension strategy is also called the X-drop method because the cells that we explore in the dynamic programming table resemble the shape of an X. Using the X-drop strategy, we end up exploring a much smaller portion of the dynamic programming table. Roughly, the amount of time we would spend to work on this problem is proportional to the summation of the lengths of the sequences rather than the product of them. In other words, the time complexity is linear in terms of the lengths of the sequences. We haven't really discussed how the locations of the seeds were found before they can be extended using the X-drop method. We certainly don't want to explore the entire table to find the seeds. That would defeat the purpose. The idea behind finding the locations of the seeds efficiently is to pre-process the database and create an index from it. An index is very much like a table that you would have at the end of a book that lists the locations of each word within the book. Let's consider the problem of finding ACC within this long sequence. We would normally have to scan through the entire sequence to find where ACC is present. But we can also build an index to speed up such searches. We would go through the sequence once and record the positions of every triplet of characters. For example, as we scan this sequence, we would see a CAC. We would go to our table and record the location 1 for CAC. And then ACT, we would go in and record the location of ACT in the table. This table that we build is sometimes called a hash table. Now, when we are asked to find the locations of ACC, we just look at this table and just read out the numbers that we have already stored. We no longer have to scan the sequence. Building the index, of course, takes time, but the premise is that the database doesn't change and we would only have to build the index once. We can then search this index many times, so speeding up each search is well worthwhile. The hash table index that I just described is used by a program called FASTA, which is another sequence similarity search method not as popular as BLAST. BLAST uses a different indexing method called finite state automata. In this method, the database is compiled into a state machine such that when we have a new query sequence, we don't have to separately ask where are the AGCs, where are the GCCs, and what are the CCAs, and so on. But rather, we take the query sequence and thread it along the state machine to find the locations of all the triplets. The transitions in this FSA are shown as black arrows with orange labels. We start from the state 0. In the query, we have an A, so we follow the A transition, which takes us back to state 0. And then we have a G. We follow the G transition, and we are again at state 0. Then we have a C character in the query, so we follow the C transition, which takes us to state 1. And then we have another C, we stay at state 1. We have an A, which takes us to state 2, and then a T, which takes us back to state 0. Some of these transitions have associated outputs, such as when we took a transition T from state 2 back to state 0, we have an output that tells us that the triplet CAT is located at positions 24 and 32 of the database. We move along this state machine until we consume the entire query sequence, and when we are done, we would know where each of the query triplets are located in the database. I'm not going to discuss the details of how this state machine was constructed from the database. regardless of whether you use a hash table or a finite state machine, searching for a single triplet in the database becomes instantaneous. Searching for all the triplets in your query would have a complexity that is proportional to the length of the query, which is a negligible factor compared to the database size. The seeds used in BLAST to initiate an extension are also called k-mers, where k represents the word length. So far, I have talked about using triplets, which means I have used k equals 3. Let's talk about how the value of k would affect the time and the accuracy. Consider using a small k, such as k equals 1. That means there would be about 1 in 20 seeds in the dynamic programming table, assuming we are using a protein sequence. And when we extend each of these seeds, we would end up practically exploring the entire table, defeating the purpose of seed and extend strategy. If we use a large value of k, say k equals 10, we would find no hits at all, missing out on any genuine high-scoring local alignments. The sequences may not share a common section that is that long. Large values of k may also require that the hash table we build is huge. For proteins, we would need to keep 20 to the power 10 entries, which would not really fit in our computer's memory. There is a trade-off between speed and accuracy when we select for a value of k. Using small k value is more likely to find the best alignments, but it also ends up taking a long time to complete. Using a large value of k means there are fewer candidate seeds, so it would take less time, but we are also risking not finding the best alignment. The value of k in BLAST was determined empirically, such that it results in significant speed up in the computation and does not miss out on any significant alignments. For proteins, the word size is 3, and for DNA, the default word size is 11. BLAST implements additional strategies to perform a sequence homology search. In some sequences, there are low complexity regions which are short repetitive patterns. Using these low complexity regions in the query would dominate the alignment scoring, and we would end up with a large number of hits from the database where similar low complexity regions are present. These low complexity region alignments are not of as much interest when we do a sequence homology search, so BLAST filters them out before carrying on with the rest of the process. Low complexity regions can be visualized in a self-dot plot, where they would appear as a band of gray-black region. When BLAST shows us the alignments containing these low complexity regions, it may replace these regions with X's to indicate that they were not considered part of the alignment search and scoring. Another strategy that BLAST implements is, when searching for a protein, in addition to using the k-mers extracted from the query, it also uses k-mers that are highly similar to the query protein. For example, given this query protein sequence, BLAST would not only consider TTC, but it would also consider SSC, TSC, and STC as seeds, because serine has a high substitution score with threonine, and they are considered interchangeable. These neighboring k-mers are predetermined from a substitution matrix, where amino acids with high substitution scores are considered interchangeable. One final strategy that I would like to mention is that BLAST does not extend every single k-mer hit. After finding the locations of the k-mer hits, it only uses locations where multiple k-mers can be found in close proximity. Let's summarize how BLAST works. The database of sequences needs to be pre-processed once. This includes building an index. For protein sequences, it would additionally include determining which k-mers are neighbors. When we are searching for a query sequence, the low complexity regions in the query are masked out, so they don't affect the alignment search and scoring. The k-mers are then extracted from the query. For proteins, this list is enriched with neighbor k-mers. The locations of these k-mers in the database are determined using the index that was created in the pre-processing stage. These k-mer locations are filtered such that only multiple k-mers in close proximity are considered as seeds. The seeds are extended using the X-drop method, where dynamic programming algorithm is applied in both forward and reverse directions. The extension is stopped when the total score falls below a threshold. High-scoring local alignments that are found are reported as results.